Good afternoon, everyone. Welcome to the Assemble Plus conference. Today is my great pleasure to introduce you Professor Matthew Sullivan from Ohio State University in the US. Professor Sullivan is a professor of microbiology and director of the Center of Microbiome Science, which he founded at the Ohio State University. He did a joint PhD at the MIT and Woods Hole Oceanographic Institution, and did a postdoc at MIT, and then he moved to University of Arizona, where he stayed for a few years, and then he moved to Ohio State University in 2015, where he is now. He's received several awards and published many papers, particularly in top journals like Science and others. He also received prizes from the Beckman Scholars Undergraduate Research Mentor and postdoctoral as well. Researcher mentor, and he's in the editorial board of Environmental Microbiology and the ISME Journal. It probably has much more than what I said, but this is what I found very quickly on the web. Certainly, we are very thankful for Professor Sullivan to have agreed to give this talk, which will be about ocean viruses, patterns, process, and paradigms for the global Tara Ocean expeditions. Thank you, Matt, for being available and for giving this lecture. I think it's going to be very exciting, and so you can start. Thank you.
Great, thank you. Thank you so much, Adelino, for the very kind introduction, and thanks for the opportunity to share some of our science with this group. It's nice to speak to a marine-focused audience. I often have to spread the marine works time with a lot of other things. So I want to share with you today the stories about viruses in the oceans, and particularly focused on a really exciting expedition called Tara Oceans that is really a European-focused expedition, and I'm one of the few US folks involved, but it's been an incredibly exciting journey. So I hope you'll really take part with me in this journey, and I hope you'll come away with a lot of new thoughts about viruses and the possibility of studying viruses in whatever your systems are. So here's an outline of where we'll go today. In brief, I'll just try to get you excited about studying viruses and microbes and mention a little bit about how we study them. And then I'll focus the talk on our oceans work, where we're studying patterns, processes, and paradigms, and some hints of some new exciting work in the RNA virus space towards the end. So why should we study viruses and microbes? Viruses usually are not that interesting to most people, although in the midst of a pandemic, we're newly recognizing the importance of viruses. But viruses are actually critical players because of who they infect. And so if we start in the human body, you have as many microbial cells as you have human cells, and there are viruses which impact them. If we look in soils, there are microbes critical for biogeochemical cycling and bringing nutrients to plants. There are also microbes and or viruses which can cause pathogenicity or disease in plants. And in the oceans, there's really important microbes that produce oxygen and as well as other biogeochemically important cycling. And half of the oxygen that you actually breathe comes from marine microbes in the sea. And my goal is, and my group is really to understand what role do viruses play in those important microbial processes. And so in the oceans, we can actually see some of these marine microbes. These are coccolithophores that you can see from satellite when they bloom, the little patterning. And these can have really important roles in the atmosphere. Here we're seeing the atmospheric carbon dioxide that cycled for the last hundreds of thousands of years from between 200 and 300,000 or 300 parts per million in the atmosphere until recently, when we as humans have perturbed this natural cycling of carbon dioxide in the atmosphere to not only get beyond the 300 parts per million barrier, but in 2015, we went beyond 400 parts per million. And the oceans actually absorb that carbon dioxide by diffusion through the air-sea interface. That leads to, through Bajerum chemistry, into carbonic acid. So ocean acidification has its roots in the carbon dioxide that we humans put in the atmosphere. And the fate of that carbon dioxide in the ocean is really determined by the plankton or the microbes that are in the sea. If those microbes use the carbon dioxide to produce more biomass that's large, it may sink out of the water column and say buy us a couple of 100 years. Or if they produce smaller biomass or remineralize that carbon dioxide, then that's going to lead to the oceans breathing carbon dioxide back in the atmosphere. This has big implications for the oceans. As I mentioned, it causes acidification, and there's lots of evidence now, both at Hawaii and Bermuda Atlantic Tide Series. And it also leads to warming of the oceans, in particular, because carbon dioxide is a greenhouse gas. It warms the surface oceans and should increase stratification of the oceans. This warming of the oceans leads to a lot more energy that's available for the oceans to the atmosphere, and that can increase the frequency and severity of storms. And all these things impact not only coral reefs, which I think we see a lot, and it's a very visual representation when they bleach or are stressed due to acidification, but the organisms at the absolute base of the food chain, including gymnosomes, pterosomes, radiolarians, and coccolithophores, all have calcium carbonate in their exoskeletons, and so are all impacted by this kind of acidification. And so I'm interested in viruses and microbes and how they may modulate that ocean carbon dioxide role and that ocean carbon buffering role. And this is sort of, when I started, the state of our knowledge. So we thought there were very few viruses in the oceans. And then several groups started to concentrate seawater and look at it under an electron microscope and eventually epifluorescence microscopy. And so this is a DNA stained seawater sample under epifluorescence microscopy. And the small dots are viral particles and the bigger dots are cells. And so what we learned is there's a lot of viruses in seawater, 50 million viruses in a mouthful of seawater. If we do tracer experiments and bottle incubations, about 1/3 of cells are lysed per day by viruses. And a big problem or big challenge is that we don't grow most marine microbes. And so therefore we can't grow most of the viruses. So very, very few of these viruses that we can see with microscopy are something we have in culture and we can do experiments with. And if you do experiments with a genetic tracer, this is one data point scaled to the global oceans. They move a lot of genes per day. So if we think about something like antibiotic resistance or the ability to use a certain nitrogen or phosphorus substrate, viruses can move that capability or that trait from one microbe to another if there's selection to do so. A lot of toxins we think of, like cholera toxin, pertussis toxin, bordetella toxin, Shiga toxin, these are not encoded by the microbe, but actually by a virus inside the microbial genome. And so when the virus has an integrated prophage, gets upset, so to speak, and gets induced, that's when the toxic outbreak happens. And a big challenge in the field was sort of translating from what I call counting dots here to ecosystem impacts. How do we get at what the ecosystem role of these viruses are? And so when I entered the field, here's how we studied viruses. First, you could use a culture of interest. If you could grow it in a lawn, which is tough for a lot of marine microbes, you could get plaques. So these clearings are where a single virus infected a cell, lytically burst and produced more viruses that infected follow-on cells. And so these clearings are viruses that have infected cells in this more turbid lawn. You could look at viral particles by electron microscopy, or you could look at them by epifluorescence microscopy. And each of these had big challenges. The first one is we can't grow most marine microbes, let alone grow them in lawns, in plates. And so that makes this very challenging. And any virus that you'd find would be only for that particular host that you're using. Of course, many of you know there's a million cells per mil-ish around much of the oceans. And so having all those types in culture would be a challenge. A second issue with electron microscopy would be you see these shapes, and the shapes are beautiful, the morphologies are beautiful, but there is no understanding what the host is or the biology of that virus. And then with epifluorescence microscopy, it's very scalable, unlike electron microscopy, but it's really just numbers of dots, and so there's no link to hosts or biology. And so really the field has shifted largely towards using sequence-based technologies, viral metagenomics, which we often call viromics. But there's a big problem with viromics too. There's actually many problems, but I'll just say over about a decade, we solved almost all those problems. But here's one that remains a challenge. So when we go out and we try to figure out who is in the water, we're looking at eukaryotic cells can be looked at with 18S ribosomal DNA. Bacteria and archaea can be looked at with 16S ribosomal DNA. And viruses themselves have no barcode gene. And so that's led to shotgun metagenomics or viromics. But if you have no barcode gene, how do you count? And so we actually first focused on this part. We can generate sequence data, but in the early years, we were using both systematic and stochastically biased approaches. And so it took us 6 years to develop quantitative viromic approaches. And I won't belabor the point except to say there's 3 ways to count in viromics-based ecology. The first one is you can use machine learning approaches with Kamer signatures, so looking at the kinds of words in the sequence space, and then use modern machine learning analytics to be able to do community ecology. You can predict genes in the assembled contigs, and those genes and their abundances can be assessed to do gene ecology. This would be if you're interested in what functions are important in environment 1 versus environment 2. Or you can do population-based ecology, and this is actually a very, very challenging and controversial part of the field, and I'll come back to that. But I'll just mention at this point that we now can look at viruses at both a species and genus level, and I use those terms very formally. I hope to convince you of that later. So viromics is ready for prime time, and I'll just mention that we've put a lot of work into making sure that all these capabilities are completely democratized. So I want to just introduce you all to iVirus if you don't know about it. This is on a publicly available cyber infrastructure called Cyverse. And you don't need to worry about the figure here. This is just the workflows of the ways that viromics is a little bit different than other flavors of metagenomics. And it's all in published papers. Why you should care about iVirus is that it's free compute at Cyverse, funded by the NSF. The documentation is extensive through live protocols at protocols.io. We've spent a lot of time benchmarking for the field viromics. I list a couple of papers here. There were 5 or 6 before these two to make sure that students aren't losing time troubleshooting every step in the pathway. We've really helped develop standards. And then we've gone out and tried to really democratize this capability by training particularly postdocs and graduate students in viromics through 6 workshops since 2016. And the next one will be next year in May. We just held the most recent one last month. So hopefully that's a little bit about why we should care about viruses. They can be accessed now, and what kinds of things are we learning in the oceans? What are the patterns, processes, and paradigms that we're seeing? So I want to introduce the Tara Oceans Consortium. If you haven't heard of it, it was led out of France, but involves a lot of countries in the EU and has at least 30 PIs at this point. And the goal was to go out and get global sampling along the cruise track that you see here, using standardized approaches, combining metagenomics, metabarcoding, metatranscriptomics across many size fractions that cover 8 orders of magnitude from viruses to fish larvae. And all this happened on a sailboat. This is the Tara, the vessel that was used, the research vessel used for this. And so it required a lot of us to completely reimagine our sampling strategies to be able to go out and get samples that could be actually pulled off on a sailboat. I'm going to focus on the virus work, but there is beautiful work for bacteria, archaea, and the microbial eukaryotes from the rest of the consortium. So if you're interested in those spaces, I encourage you to have a look at those. So what did we observe for viruses? I'll just tell you, when we started, 90% of what we observed was completely unknown. So we would survey sequence space, and it was unlike anything that was available in the public databases. And this not uncommon to this group. Most of you know that as we explore ocean space, it's underrepresented in public databases. And so a lot of it's unknown. So our first role really was developing catalogs. So when we started, there were about 2,000 viruses of microbes that had been sequenced for genomes. And so those are represented here by two pictures of viruses, where each picture is 1,000 virus genomes. When we first got into this, we augmented those catalogs about 15-fold, and we've since augmented those about 100-fold to where now we have a catalog of 200,000 virus species. This was quite challenging because, as you saw earlier, most of these are new. So we had to develop a machine learning analytic even to establish basic taxonomy. And so this uses gene sharing networks to reimagine a systematic and automated way that's scalable to be able to classify these viruses. So this has actually now been heavily interrogated by people at the International Committee on Taxonomy of Viruses, and it's stood out to be robust. In fact, it's helped us identify at the ICTV places where our taxonomy was not systematic and was skewed by personal preferences one way or another. And so this might be an emergent way for ICTV to better classify new virus sequence space. So if we have these catalogs, what can we learn about how the oceans function? So this is a cruise track of the Tara, where each number represents a station with a gray dot, where the boat spent about 24 to 48 hours sampling. And then the arrows, the colored arrows, represent the genome-enabled virus tracking. So the flow of virus populations at different depths that are listed here in the bottom left. And overall, what we see when we use these genome-enabled virus tracking is that the viruses follow the major ocean currents. And this is not surprising. Of course, viruses don't swim, and so it makes sense that they're passively riding with the major ocean currents. But I wanted you to just stick a pin in that, and we're going to come back to that a little bit later for another pattern that comes out of this. Beyond just tracking flow of viruses, we could identify which viruses are even out there. And so when we did this, one of the major, actually the most abundant virus genus was the T4 viruses. And these are the basis of molecular biology. If you use any PCR reagents, if you use genetic and cloning reagents, a lot of those derive from a virus like this that infects E. coli. And so we've spent time unpacking what makes a ocean T4 different from a sewage or a gut T4. And so there's a lot of beautiful comparative genomics work following the difference between ocean cyanobacterial viruses and your gut viruses that are the same genus. The third most abundant genus was Phi-38-1, which nobody had heard of. And this particular virus, we happened to work hard and got it into culture, and it's now probably one of the most well-studied viruses on the planet because of some work led by Christina Howard-Verona. So we actually have maps and names now for the most abundant viruses and can use those to test hypotheses that come out of the viromics. So with catalogs and these maps throughout the oceans, we're now trying to use these data to be able to improve climate change impacts or modeling of climate change impacts on ocean ecosystem services by looking at diversity anomalies through time, adjusting how we might think about fisheries catches, looking at marine protected areas. And recently I was just in Paris at the Green Ocean Modeling meeting where we're thinking about bringing viruses into the climate models, which inform the IPCC and their eventual climate predictions. So those are the patterns that we observe, and I could go into a lot more depth on many of those. There's fundamental ecology at many levels, but I want to hit some of these other points too. We next look at processes that viruses may impact, and the interest in this was that in cyanobacterial viruses in culture, we had seen that viruses encode photosynthesis genes. And so these are core photosystem genes which directly impact photosynthesis. And that led to the idea that viruses could have auxiliary metabolic genes, or AMGs, and that these could be direct ways that viruses reprogram core metabolisms. This was studied extensively in the cyanobacterial culture-based literature. And so I just want to overview this idea. Why is it that viruses have photosynthesis genes? The idea is that in the cyanobacterium, you have a photosynthetic membrane with two reaction centers, PS2 and PS1. And light strikes PS2, where a photon of light excites an electron, enables PS2 to be able to split water into hydrogen and oxygen. And there's been 4 Nobel Prizes that have been won here studying this process, because oxygen and hydrogen are, of course, critical. That excites an electron through a series of electron donors and acceptors to PS1, where a second photon of light strikes, excites an electron, and that gets passed to ferredoxin and onto biomineralization reactions. The viruses are probably randomly sampling the host DNA, and those genes which improve viral fitness stick. One of those genes that sticks or fixes is PSBA. And this is the gene which encodes the core reaction center protein in PS2. And we know from culture-based work that if you turn off the lights or you poison PS2 with DCMU, then you don't get cyanobacterial viruses. So we know that cyanobacterial viruses require photosynthesis to be able to make new viral particles. We also know that PSBA is replaced every 30 minutes, and this is because of that oxygen radical formation here can damage that protein. And we know that viruses, when they infect the cell, are going to shut down the host transcription and translation capability. And so that means that the virus wants photosynthesis to continue, that PSBA is damaged, and you shut down the host's ability to replace it, then the virus carries its own PSBA that can come into the photosystem to be able to help keep photosynthesis going. And there are other genes, other photosynthesis genes that viruses also have. If you're a broad host range infecting virus, you might have PSBD, and that's probably because PSBA doesn't work with some of the hosts. And so you have to make sure that you have a PSBD, a heterodimeric partner, which will keep a functional PS2. They may also have plasticinin or ferredoxin, and these are rate limiting steps of electron flow away from PS1 or PS2. And so they may prevent damage. And then highlight inducible proteins, which are thought to stabilize the PS2 reaction center. So viruses are probably randomly stealing host genomic DNA because they're sloppy at points, depending on the virus type, and how they incorporate new DNA. And a subset of those photosynthesis genes out of maybe 80 photosystem related proteins, they only have a handful that have been selected for and fixed in virus genomes through time, presumably because they improve viral fitness. Now, when you get beyond cultures and you use viromics, you can screen and find there's 35 central carbon metabolism genes in viruses spread throughout the Pacific Ocean. And no virus has all of these central carbon metabolism genes. They only have a few. There's also, in the rest of the oceans, sulfur cycling genes, nitrogen cycling genes. And so there's lots of different ANGs that viruses have stolen through time and lead to direct links for who their hosts may be, as well as an insight into the kinds of metabolisms the viruses may be directly manipulating. And a big cautionary tale is that viromics is never pure. When we create some purified viral particle concentrate, there are 2 ways contamination can happen. One is that something in the process wasn't perfect, and it rarely is perfect. And the other is that gene transfer agents or capsid encoded DNA that is just cellular DNA can get in the mix because they actually would be properly purified with the viral particles. And so this has led to some really poor estimates of AMGs where cellular DNA is being mistaken as viral. And so when you get a virome and you look at it and you see the amazing metabolism gene of interest just make sure it's really on a viral config and not on a cellular config. And there's some advice for how to do that in a recent piece of work from Ajit Pratama, where he's providing a standard operating procedure. And there's also an automated way to do this through Kelly Wrighton's DRAM-V tool. So beyond patterns and processes, what kinds of new ways of thinking are coming out of viromics? So I want to walk you through 3 paradigms. There could be another half dozen here, but I'm going to walk through 3 paradigms for how viromics is changing our viewpoint. And the first relates to virus sequence space. So when we think of viruses, we often think of them as being fast evolving and subject to rampant mosaicism. And so it really calls into question whether we should even be able to see populations in environmental data. And so what this would look like would be if you had gene flow or rampant mosaicism or gene exchange across viral types that's really high, then we would expect the plot on the left to represent viral genome sequence space, where the yellow viruses blend into the light green viruses, blend into the dark green viruses, blend into the blue viruses. So this would be a continuum in virus sequence space whereby you couldn't really draw a circle around any of these groups and say that's a population. And the alternative would be that there's a higher rate of gene flow within a group than between groups. And in that case, yellow viruses should look a lot more like yellow viruses than they do the green or light green viruses. And you might see discrete populations in sequence space. And again, I think when we think of viruses, we think of high mutation rates, but I will just posit for you that actually most of the data we have on gene flow and mutation rates are from single-stranded DNA and RNA viruses, because these infect humans. And these are the ones that virologists have studied for a long time. And those rates are actually orders of magnitude higher than they are for double-stranded DNA viruses. And so maybe there's a chance that there's discreteness in virus sequence space. So to address this question, I went out naively for 10 years, isolating cyanobacterial viruses here in gray on many different hosts for many different environments. So this is my PhD and postdoc work for nearly 10 years that resulted in these gray dots. And this is now a 3-axis virus sequence space being plotted here. And so it's hard to tell, are those clustered or not, right? So we wanted a higher throughput approach to be able to see a lot more data at once. And so we developed viral tag metagenomics as a way to capture in a very, very fast way viruses. And so this is to look at 10 to the 7th viruses in a 10-minute experiment. So a ton of sequence space all at once. So what we did first was just go to seawater and isolate some viruses. So in blue are 4 viruses isolated from a particular seawater sample. This is off line 67 off of coastal California. And what we see in blue is that 2 of these viruses look very different from each other, but another 2 look nearly identical to each other. So again, I'm left unsure what sequence space looks like. But when we did our viral tagged experiment in red dots, we got all these viruses, which shows up as much diversity as I'd found across many hosts and many seawater samples in 10 years. We got out of a 10-minute experiment. And then when we resampled the data to create in silico versions of those, we found the clouds around the red. And in all cases, those are distinct populations. And in the few cases where you think they're on top of each other or maybe overlapping, the end of the page axis here that's shadowed down below show you that those are clearly separated on the 3rd axis. So those populations are separated by about 5% average nucleotide identity. And we've gone on with proper population genetic metrics to formalize this. So if we actually go to the same seawater sample, we isolated 142 cyanophages, sequenced all their genomes, and then applied population genetic metrics. What we learned is these populations are species. By that, I mean that gene flow is higher within and between these populations. and that the populations are under differential selection. So there's clear niches for these different populations. And then as well, when we look in other heterotrophic bacterial viruses, so pseudo-altermonophages, we can actually look at host ranges and show that their host range is concordant with the population-based definitions that we've established. So virotag metagenomics and viromics helps us assess and show that viral sequence space, at least for DNA viruses, is clustered, and that those populations that we see emerging from that are species-level entities. So the second paradigm that viromics has been informing is related to the ocean carbon pump. And I mentioned at the beginning, the oceans are this critical player in carbon flux and in helping have climate change due to pulling down about half of the atmospheric carbon dioxide. And I also mentioned earlier that we'd tracked genomically viruses along this transect, and that led to us realizing that viruses ride the ocean currents. But I said, wait, we'll come back to that. And what I didn't tell you earlier was that we also saw at each station these X's. And so I'm just toggling back and forth. The X's are where viruses appeared to sink at each station. Now, that doesn't make any sense because viruses shouldn't sink. They have a low Reynolds number. So what could possibly be happening? So the paradigm was actually that of the virus shunt. So the idea was that bacteria and phytoplankton get infected by a virus. That leads to particulate and dissolved organic matter that's rapidly recycled by heterotrophs through this process called the viral shunt. And this was really the paradigm in the field since the late 90s. But Marcus Weinbauer suggested in 2004 that maybe that local process of cell lysis could also lead to really sticky, larger aggregates, which could sink out of the water column and lead to carbon or cellular export. And in other papers, there was suggestion that virus-infected cells might be preferentially grazed, which would lead to fecal matter sinking out of the water column. So these are different ways that viruses could sink or appear to sink in our sequence data sets. And so we wanted to test this with the genes to ecosystems modeling approach that Tar Oceans had developed, whereby sequence data were converted into biodiversity numbers, which could be run through an ecosystems model that had both machine learning components and ecosystem rules to try to assess this question. So this is, again, the Tar Oceans transect, where the white dots are 24 to 48 hour stops for the Tara. And now we've got a depth profile into the ocean. And the colors here are blue, where there's areas of low carbon export, and red or yellow, where there's areas of high carbon export. And so Tara used a very novel approach, an imaging-based particle and size-based approach to estimate carbon flux throughout the surface oceans. And then we layered all the Tara biodiversity data from viruses to fish larvae on top of that, and used this machine learning plus ecosystems modeling approach to ask which organisms are driving carbon export in the oceans. And the answer, by far better than the other organisms, was viruses. And this was exciting because virus abundances alone could explain 89% of the variation in carbon flux throughout the global oceans. And that's a big deal. When you're looking at ecology, explaining 89% of the variation of anything is very, very uncommon. This also lent support to the idea that viruses might be really forming these aggregates and exporting them out of the oceans, if they're the big driver of carbon export. And it also has led to follow-on experimental work where we're using stabilized tope probing to quantify the shunt versus the shuttle. The other point I want to make is that these are really generalizable capabilities. So if you're not yet using, I call it the Guidi et al. WGCNA and leave one out methods, this is really important for looking at any kinds of ecosystem features if you have large scale omics data sets. So the third paradigm is one of virocells. And so the virocell was introduced by Patrick Fortier in 2013. And the idea is that when a virus infects a cell, that cell is really no longer acting in the way that we would consider it being a cell. It's acting as a viral factory or a virocell. And so a virus-infected cell is a virocell. So what we were interested in exploring was what happens to a cell when it's infected by a virus, and then actually by a second kind of virus in an independent infection. And so we use time-resolved multi-omics analyses to look at this, and we have data from that to be able to quantify what the uninfected cell looks like with respect to energy, metabolic functions, and uptake and output of resources of different types. And so this is just a schematic, high-level picture of that to show you what the uninfected cell might look like. And when we infected it with a virus called HS2, the HS2 virocell had more energy required, more diversity of metabolic functions, particularly brought in certain kinds of resources, and that led to a large viral fitness of this virus. When we infected by another kind of virus, a potovirus, HP1, the HP1 virus cell used even more energy, had more metabolic functions, and imported a lot more resources because of a genomic mismatch between this virus and the cell. And so what it means is that if you're doing a 16S screen of this particular sample, one of those 16S sequences would be behaving as an HS2 virus cell. Another 16S sequence would be behaving as a HP1 virus cell. And the third one is the only one that would be behaving in the way that you think of. And what's important to remember is that about 1 in 3 cells are infected by viruses. So I think the virus cell idea and the fact that the virus-infected cell is a completely different entity from the uninfected cell is really critical as we think about ecosystem modeling moving forward. So those are 3 paradigms that we've begun to unpack. And I just wanted to pause and say, everything I just told you is actually just DNA viruses. And there are other kinds of viruses in the sea. And so in particular, RNA viruses, and these might be a lot more important for eukaryotes, for those people that are interested in microbial eukaryotes in the oceans. And we've recently had a large contribution to the RNA virus space through taking those same global oceans data sets from Tara and trying to understand, on the one hand, their evolution, and on the other hand, their ecology. So I'm just going to summarize those in one slide for each of these papers. So the idea was, first, we actually had to improve analytics. Turns out these are 3 billion-year-old proteins, the RDRP protein that's used to capture these RNA viruses. So we had to improve analytics to even see these better. With that improved analytics, we then screened 28 terabases of data from Tara Oceans to find 44,000 RNA virus contigs, and this led to about 6,000 complete RDRP signatures that we could use. It turns out that the field is full of controversy about how you can even align these very, very old proteins, because they're so old that it's difficult to decide on what are real homologous positions. And so we decided to try to get away from a subjective manual curation of alignments by using a machine learning approach, and tune that machine learning approach to actually see if we could recapitulate the ICTV's, the International Committee on Taxonomy of Viruses, official taxonomy, and we were able to get 97% concordance. And this led to a systematic and objective pre-filter to allow us to really improve the global alignments for these trees. And when we made our global tree, it led to this picture. In this picture, what you're seeing is, in gray, all of the known RNA virus phyla. So there were 5 known RNA virus phyla, and that's from all of previous work. And in the more reddish sequences, you're seeing 5 new phyla that emerged from the global oceans data. So the oceans themselves had RNA viruses from all 20 known RNA virus classes, about half of the known RNA virus families. But even though these are relatively known, 99.7% of these were all new species. So even amongst the 5 known phyla and their classes and families, the ocean ones were still quite different. They represent new species. But beyond that, the ocean data informed us about 5 entirely new RNA virus phyla. So that allowed us to see that much further into RNA virus sequence space. And I'll just say that, again, these global alignments are tricky. So we spent a lot of time looking at those phyla to see if they had additional support. There's 3 flavors of additional support that are consistent with these being phylum level taxa. And I'll just flag that in honor of Tara and Tara Oceans, we named the most abundant of these ocean RNA viruses the Tara viricoda. And not only were they widespread across the Tara Oceans data sets, but they also, when you look at their early branching evolutionary traits, they look like they're right at the origin of life. So maybe the missing link between a capsidless and a capsid containing nucleic acid. So a really exciting early evolutionary story. The other half of that is to take all those new RNA viruses that we've now carefully classified and look at their fundamental ecology. And the first step was defining a species unit, which is highly controversial in RNA viruses. So what we did was define it, and then we looked carefully at many different ways you might define it around that to look for robustness of the following observations. And they're all very robust. So our species unit is robust for doing ecology. And the things that we learned were that there's structure across the global data set, such that there are 4 meta communities or ecological zones. We can begin to see clear depth patterns. In green are the RNA viruses, starting with high diversity based on Shannon's H in the surface waters and decreasing with depth. And that paralleled largely what we saw with DNA viruses. A surprise was that RNA viruses show very little latitudinal diversity gradients. So DNA viruses, if we look from the left, which is the South Pole, to the equator, to the North Pole, DNA viruses showed the typical hump until we get to the North Pole, and then there's this biodiversity hotspot in the Arctic. And we saw very little evidence of a latitudinal diversity gradient for any of the RNA viruses. And I will flag these are RNA viruses from metatranscriptomes, which means only from the cellular fraction. So we're not looking at RNA viral particles. We have from these data mapped out ecological drivers and assessments of community structure and compared those to both the DNA and RNA viruses, and the details are in the paper if you're interested in that. We also saw auxiliary metabolic genes or key metabolisms that the RNA viruses seem to be directly manipulating. And for carbon flux, the RNA viruses are also highly predictive, just like the DNA viruses, and they seem to infect key carbon cycling organisms critical to the biological carbon pump. So I think RNA viruses are also playing a really critical role in ecology, and we hope that these analytical steps that we've developed to create these RNA virus catalogs will be helpful for researchers to do follow-on work. So just to wrap up what I've told you today, when I started, we knew that viruses were abundant, about 1 million to 100 million per mil of seawater, and we counted them as dots, which is the way I'll describe the cyber green stained dots in the microscope. And we now have an approach that is quantitative and systematic to look at their genomes. We can use those genomes to establish catalogs, and we have 200,000 species of DNA viruses, as well as about 7,000 species of RNA viruses now in hand. Sorry, 5,500 species from 7,000 complete RDRP sequences of RNA viruses. And to get beyond species at genus level taxonomy, we've developed gene sharing machine learning network analytics to be able to establish that. And we're trying to prove that to actually be all of hierarchical taxonomy. We've observed many auxiliary metabolic genes. I flagged the idea of photosynthesis genes, carbon cycling genes, sulfur and nitrogen cycling genes. There's also phosphorus metabolism and uptake genes, et cetera. We use these catalogs and population-based ecology to assess through machine learning and ecosystem modeling and genes to ecosystems approaches which organisms best predict carbon flux, and that was viruses. And I didn't mention it earlier, but not only can we see viruses are the best predictors of carbon flux, but we know which viruses matter. So this is the variable importance in projection. We can tell you which viruses are most important in those predictions. And this really establishes population-based viral ecology for DNA viruses. And I will say all that is wrapped up in a toolkit that's available publicly, along with many resources at iVirus, which is publicly available on the NSF cyber infrastructure, CYVERS. I then said it's not just DNA viruses, there's also RNA viruses, and surveying the global oceans let us see 5 additional phyla beyond the 5 known phyla of RNA viruses. And this has really been a basis to look at their ecologies, show that they also have cool metabolisms that they're tying into, and that they infect critical players in the biological carbon pump and strongly predict carbon flux in the oceans. And this is some of our oceans work. We also do work in soils and in humans, and those biomes allow us to really cross in a lot of ways and learn from the complexities and universalities of these different systems. And so it's a really vibrant and exciting research space. I've been very fortunate to be funded by a number of programs, including the Gordon and Betty Moore Foundation, the Joint Genome Institute, DOE and National Science Foundation, as well as the DOE EMSL services program. And if you're excited about any of this work, I do have several positions from some new grants that have just been awarded for postdocs, graduate students, and in the Center of Microbiome Science, we're looking for a second data analyst to build out our microbiome platform, which is serving 109 faculty at Ohio State. Thanks for your time.
Thank you very much, Matt. Fascinating and very exciting. All this this world. Is there any questions from the audience? Oh yes, Nick, go ahead.
Thank you, Adelino, and thank you, Matthew, for a really, really interesting talk. That was fascinating to look into. My question is something you mentioned a bit about midway through your talk when you were starting to talk about the Tara work and looking at the viruses and also on the predictions around carbon. And it made me wonder, we're doing a lot of work now on setting up genomics observation and so on. And there's a lot of thinking about what would be, good indicators, what would be worthwhile looking at, what should we be communicating to our policymakers and so on. Based on your work and what you're seeing, do you think that viruses would then be a good candidate for being one of these sort of indicators that would actually give us an awful lot of insight into what's happening in terms of carbon, the health of our microbiomes, and so on?
Yeah, and I'm beginning to wonder if we should take it a little bit further. I worry that we as a global society have really failed on this sort of consumption and output of carbon to where we're really shifting the ecosystem towards a dangerous tipping point. And if we get over that tipping point, it might require some drastic solutions. And so I'm trying to think about if viruses are such a big predictor of the biological carbon pump, can we use them to actually tune the knobs of this great ocean system? And so certainly viruses can be good indicators and help us study and think about key spots in the ocean ecosystem, but they may also be a tool in our toolkit down the road. And so I'm trying to get the science out ahead of that enough that we could make meaningful advice, because I think we all know that the second that we humans geoengineer something, all hell breaks loose and there's all these unintended consequences. So I'd like to get ahead of some of that and see if we can at least develop some solutions in the near term. There's lots of ways you can imagine biomarkers and predictors coming out of these sequence-based data sets. And I think that's been underexplored. And I would encourage people to look into there's a lot of Tar Oceans data. I mentioned the 2 papers on RNA viruses. That's from 2.5-year-old published data. And so there's just really rich stories that can come out of the Tara data and biomarkers at a global level and then complemented with marine lab specific experiments and would be really beautiful. Does that answer your question?
Yes, absolutely. Thank you very much.
Sure.
Okay, Matthew, thank you. Babak, do you want to ask your question? He's got a question in the chat.
Hello. Yes, I can. Maybe it's better to ask. Thank you for your nice presentation. I just have a doubt. If I grasp well from your presentation, you expressed that viruses may also need cyanobacteria to be alive, right? So could they affect carbon import as well, like the carbon export that you explained? I'm not sure if I grasp well that part, so that's why I asked this question.
Yeah, so I'll see if I can. So cyanobacteria and photosynthesis continuing is required for cyanobacterial viruses to be produced. And so the virus, when it infects a cyanobacterium, is taking over that cell's capability for photosynthesis. And so there's actually a couple of research groups asking the question, does that virus-induced cyanobacterial photosynthesis differ from cyanobacterial photosynthesis? And so I think there'll absolutely be some differences. There's no pest domain in the viral version of the PSPA. Sorry, there's a pest domain, but it's mutated. And so this is the part that makes that protein turn over rapidly. So the viral version doesn't turn over every 30 minutes like the host version does. And you can imagine that that impacts photosynthetic efficiency, et cetera. So there's beautiful biochemistry and in vitro work that could be done, as well as physiological work that could be done studying those cyanobacterial photocells.
Mm-hmm.
Okay, thank you so much.
Sure.
I have a naive question. From what you showed, it seems as if the viruses tend to hijack, let's say, the whole, several of the host's proteins or whatever. And so my question is to what extent there is specificity? I mean, in terms of course, viral species versus host species. Is it just a broad kind of functional thing, or what do we know now? I mean, in terms of specificity.
Yeah, no, great question. I didn't get a chance to go into this, but the specificity is pretty high. So in fact, we've shown in Cellulophaga that 2 cell types that are co-isolated from a seawater sample that have identical 16S sequences are infected by viruses in totally different ways. Mm-hmm. And so the 16S signal would tell you, oh, there's 2, right? But the viruses actually see at a higher resolution. And that's because 16S is an ancient, slow evolving evolutionary trait, really. And virus infection is an ecological, faster evolving trait. And so those 2 traits are just out of sync. So 16S matched the virus infection dynamics almost doesn't make any sense at all because those timescales are so far off. So yes, those metabolisms are probably tightly to a subset of what we would normally call a species, because the virus sees cells at a subspecies level. For the most part, there are broader host range viruses which would have a broader metabolic impact, but.
Okay, thank you.
Sure.
No more questions? Well, if not, thank you very much, Matt. This was a very, very interesting presentation of your work, and certainly it's a very expanding field. When we started the Assemble project years ago, we had a team from Montpellier. They were in the medical school, so they were not really marine biologists. And so they went to Chile, and basically they did what you said. Instead of using, sieves and stuff, they just went and took the water, and they discovered megaviruses. So that was one of our first really interesting discoveries out of this project, and many others have since happened. But it's very interesting. So thank you again. Thank you very much. And well, we'll probably invite you again in the future, and perhaps we can collaborate somehow.
Sounds great. Yeah, we love to be involved in any cool systems where viruses may play some role. So, and I didn't actually bring up the giant viruses because there's a TAR Oceans coordinator, Hiro Ogata, who's in charge of all the giant viruses. So I tend to focus on the other side. Yes, the giant viruses are a whole nother fascinating toolkit. So I'm glad you brought that up.
And the story of the discovery of the megavirus is a book story. So it's very interesting.
Yes, beautiful.
Okay, great.
Thank you.
Thank you again, and bye-bye.
Hi, thank you so much.